right, with the tables configured this way, I mean, there will be times, and I mean, I've got you seated facing one another so that there are definitely times that you can work together as a team. There's going to be times when I need your attention towards the front. That's why I tried to arrange these tables so that you could do both things. You could work as a team. You could also give attention to the front. As a matter of fact, there is going to be a time more towards the end of the period when some of the product of what you've done as a team, one of you are going to be bringing it up here as we try to start building a little protein molecule, a little one. So let's get the polysaccharides out of our system, and then we'll go on. So we ended, just real quick review, we ended on Tuesday talking about 3 important disaccharides, and they were sucrose. So the monosaccharide and the disaccharide, you really can't tell the difference by name. You can kind of tell they're all saccharides. They end in OSE. Glucose, monosaccharide. Sucrose, it's a saccharide, but it's made up of glucose and fructose. Lactose, made up of glucose and galactose. And I mean, you can see when we start talking about diseases like galactosemia, that these things, although they seem like they're very subtly different, they really make a big difference. There are some people who cannot metabolize galactose, and it results in galactosemia, and there's just a very subtle difference. Galactose has the OH above the ring, glucose has the OH below the ring on carbon number 4. Seems like a trivial difference, but the body doesn't think it's trivial. There's certain proteins, actually, that are needed to metabolize galactose. And if your body can't make that protein, it's not going to metabolize it. And so then it builds up to a point where it becomes toxic. So that's lactose, galactose and glucose. And maltose, look at maltose, alpha D-glucose and beta D-glucose. So it's 2 glucose molecules coming together to form maltose, but you got the alpha and the beta. And again, you'll see me, I mean, you're hearing me just express my amazement at how, I mean, I've done a lot of bench chemistry. I've done chemistry at the bench, mixing things in flasks. And so I appreciate how well living systems do chemistry. They do it really well. I mean, if it needs an alpha D-glucose, the living organism is going to make alpha D-glucose. The chemist at the lab bench is going to spend months trying to do it and get a 50% yield, and some living organism will do it 100% of the time perfectly. So it's amazing. It amazes me. I'm impressed with living organisms and the way they do biochemistry. Okay, into polysaccharides. That's another disaccharide. Polysaccharides, we're talking about, 25, 30, 50, 100 or more saccharides together. Okay, starch is an important one to think of. The storage form of glucose found in plants, so potatoes and sweet potatoes and different things, that wheat, you get wheat starch, potato starch, rice starch, so that's a polysaccharide. So yeah, the primary storage, primary means of storing glucose for plants is starch. I'm going to only point out a couple of these. Glycogen, all right? Major glucose storage carbohydrate polysaccharide in animals. like us. The glycogen is in the liver and in muscle cells. You're piecing together lots of glucose, storing it there. And then the body has the ability then to clip off glucose off of that glycogen when needed. Your blood sugar starts waning and your body will release glucose from the glycogen. You have an emergency and you need to run somewhere really fast, it's going to start, it has some in the muscle cells to provide energy. Okay, here's glycogen. So these are all glucose, individual glucose molecules. There's some branching in glycogen. All right, cellulose. This is the most abundant organic material, actually. The most abundant organic compound in nature. Cellulose. And I think I mentioned this on Tuesday. Tree trunks, stems of plants, corn stalks, the stuff that holds plants up and together and gives them structure is cellulose. We don't digest cellulose. Cows can digest cellulose. They can chew the grass, and they got a very complicated digestive system, and bacteria in that digestive system can break down the cellulose that cows eat and digest it. But to us, that doesn't mean we shouldn't eat it. This comes in the category of fiber in your diet. You know, the doctors are saying you should have some fiber in your diet. What are they saying? You should have some stuff in your diet that you can't digest. Your digestive system needs some of that material just to carry everything along through the digestive tract, to have probiotics, have a place to lodge and work. And so you need fiber in your diet, in your digestive system. And this is fiber. A little kid, and hey, eat your celery. Why am I eating the celery? It's fiber. It's mostly fiber. Is what celery is. It's mostly cellulose. Maybe a little bit of nutritional value, but not a great deal. There may be a few vitamins and different things in celery, but that's cellulose. So the three that I mentioned here are starches, glycogen, cellulose, are polysaccharides. So, all right, here's a structure of cellulose. I mean, again, I'm amazed at try to hold your arm out for 48 hours, okay? Your arm will start getting tired. Live oak tree out here holds the limb out for ever and ever and ever. How does it do that? Like this. The cellulose is cross-linked. You know, if you were making an erector set with some sort of building block set, and you know, you linked things together, cross-linked things, you'd start making a strong structure. I mean, if you just stack things straight up in the air, eventually it'll fall over. But if you stack them up and have branches coming off of them and branches coming to other stacks, all of a sudden you feel like, oh, I've got a structure here. I've got something that can support some weight. It has some mechanical strength. That's what the cellulose is. Obviously, the live oak trees out here have some mechanical strength. They're holding tons, you know, hundreds of pounds of weight out all the time. And it's the cellulose structure that has that mechanical capability built into it, okay? So that's what you're getting, and that's through cross-linking. If the cellulose was a linear polysaccharide, you know, the stuff would just fall down. It would be like spaghetti. But it's not like spaghetti. It's all bound in and cross-linked. All right, don't worry. You'll see these slides. Don't feel like you have to understand these at that level, that I got a carbon 1 to 4 bond and all of that sort of thing. know these three major categories of polysaccharides. Know what glycogen is. It is your primary storage molecule for glucose in your body, in the liver and the muscles. And then blood type. Blood type. That factors into chapter 16 because these antigens, these different antigens, type A, B, type AB, type O, are really, these are oligomers. Okay, remember oligomers are 3 to 10 saccharides. And so these tiny little chains of 4, 5, 6 saccharides attached to the hemoglobin is what is typing your blood. It is the indicator, the signal to other things in your body that make this compatible with your body, your blood type. Type O. So the green one, let's see. Well, we've got here galactose. I don't know if that's supposed to be fructose or if that's another sugar, to tell you the truth. But at any rate, there are 4 different monosaccharides building up to make that antigen. All right, so let's see here. Type AB. What's type AB? It has A and it has B antigens on the hemoglobin. So let's see, the universal donor is who? O. O is the universal donor. And is AB the universal recipient? Great. I needed a little help there. Okay, so this is the universal donor. This is universal recipient. Yeah, so type AB can handle type A, type B antigens. It can handle those. Now, why can it handle type O? I'm not sure I can answer that question. That is different. It's a little different. But apparently can accept type O. All right, so that's your blood typing. Starch, energy storage in plants. Glycogen, energy storage in bacteria and mammals. Cellulose, structural in plant cell walls. And the most common. And those are the 3 that I would say to concentrate on. A slide with just some summary terms, key terms from chapter 6. Aldose, ketose, chiral, Fischer, Haworth, stereoisomers or optical isomers. Saccharide, monosaccharide, disaccharide, polysaccharide. Reducing sugar. Did we? I know we looked over the Benedict's reaction, Benedict's reagent, and the Benedict's reaction in chapter 13 when we studied aldehydes. We didn't go over it in this chapter, did we? We should have. Is there a slide on it? Just remember, there isn't a slide on it. Slide 32? In here? Okay. I'm not seeing it. Huh. Oh, reducing sugars. Okay. Reducing sugars. Well, that is worth remembering, is being reminded of. Reducing sugars, they're going to react with, okay. This has to do with the Benedict's reagent. And before we talk, glucose, before we talk about fructose, remember that glucose plus copper plus 2, which is blue, this should bring back some memories. Copper nitrate, copper sulfate, blue. This is an aldehyde. Glucose is an aldehyde. What happens? The glucose gets oxidized. Okay, aldehydes get oxidized to carboxylic acids. And the copper gets reduced. Remember, started out blue, turned red. If you don't remember that, look back at chapter 13, and that's where I have 2 slides that show it real clearly. I thought they were here also, but they're not. And we won't waste time to go back there. But chapter 13, that was where we said you could take urine analysis. You could do urine analysis with Benedict's reagent and detect glucose in the urine by this simple little test. You add a little copper sulfate to the urine analysis, and if the blue copper sulfate turns red, it would indicate you have a reducing sugar. Reducing, that's what they're meaning by reducing sugar. It's reducing Benedict's reagent. It is being oxidized, but it's a reducing sugar. And theoretically, it should only happen with aldehyde functional sugars. But tricky little fructose, which is not aldehyde functional, it's a ketose, can go through a series of adjustments here to become aldehyde functional. This is starting with fructose. It's going through this enediol formation. So the carbon-oxygen double bond is reverting to a carbon-carbon double bond and 2 OHs. And then it's climbing on up to be a carbon-oxygen double bond on carbon number 1 and an OH and a hydrogen back down here. It's turning into glucose. Fructose is going through those adjustments, those changes, yes. No, I'm saying this is being reduced. This is reduced. Okay. This is being oxidized. So that is reduction. And that's why we're saying this is a reducing sugar. It's not being reduced, it's being oxidized, but it's reducing copper. It's the reducing agent that got oxidized. And copper is the oxidizing agent that got reduced. Okay, there's that confusing way of talking about these things in redox equations, redox reactions. But yeah, it's a reducing sugar because it can be oxidized itself. +2 is going down to +1. That's lower oxidation number, reducing. Okay, that's all I need to say about it. But just what we talked about in chapter 13 is relevant here because Benedict's reagent is used to determine if a sugar is a reducing sugar or not. Will it turn blue copper into red copper? Okay. Any questions on chapter 16? So we'll move on into chapter 18. I'm going to